Hi, my name is Robert and my protein is Cras. Cras is a small GTPase that acts as a molecular switch and plays a crucial role in cell signaling pathway that controls cell growth and division. The primary location is in the inner surface of the cell membrane, and the mutation in Cras are closely linked to various cancer. One common mutation is the substitution of glycine and cytosine at position 12, which is also known as the G12C mutation, and this mutation causes uncontrolled cell growth. The overall 3D shape of Cras is globular with regions for GTP and GDP binding and hydrolysis. It has flexible region known as the switch loops, and then these region depends on whether or not the protein is bound to GDP or GTP. And then to mimic the 3D shape, I created a globular structure with toilet paper roll and pipe cleaners, so it could like represent the flexibility in the switch region. So within the structure, the primary structure is a linear sequence of amino acids, and then it could be seen. Oh, I'm so sorry. It could be seen in the pipe cleaners, like if you like zoom in, if you could. And then the secondary structure region is the alpha helix and beta sheaths, shown like also, like I tried to like maneuver to make it look like it has alpha and beta sheets. And then the third structure is like this actual globular structure, and then it does not have four primary structure, the quaternary structure. So for the G12C mutation, replaces the glycine. I represent it by this red area. This red area, but in the normal one, it wouldn't have this red area. I just put it in just so like you can see the mutation. It can form disulfide bonds or interface with protein flexibility and conformation. I couldn't really do that on here. I just show like a location where it would have the mutation. And then this is like a whole protein of Cras. I use toilet paper, the pipe cleaners, and then another pipe cleaner to represent the mutation spot. So the G12C mutation reduces protein ability to hydrolyze GTP into GDP, meaning Cras remain active for a longer period. And this constant on signaling, it promotes the unregulated cell growth and division, which ultimately leads to cancer. And then the mutation in the position 12 is located near the binding site, and like I said, it's marked near the bright red color. There are many limitations to my models. The scale, it's very upscale, like usually you wouldn't be able to hold it. And then the movement, I can't really show between like the active and inactive. And then like the complexity, I'm sorry. The complexity of like the actual real, compared to like one you can just find online. You can actually like see it move around, like with the bonds and like the amino acids and stuff. So, but this is my protein model for Cras.